Hi, Michelle Glass here. We are back with our third episode of our Chapter 3, Lecture 3 series. We are still, remember, talking about our cell membrane, and we are talking about how material makes its way across the cell membrane, whether it's going in or out of the cell. Remember, we had our last video on diffusion, and our topic for this presentation is the special type of diffusion, which is called osmosis. So when we talked about diffusion last, we left things here where we had simple diffusion is taking no energy, it's not using any proteins, that's just the straight up movement across the membrane through the phospholipid bilayer from a high to a low concentration. With the channel mediated type of diffusion there, we're still going with the concentration gradient. So again, no ATP is required, but here those ions and polar molecules will not go through the phospholipid bilayer and have to go through protein channels instead. Then we have our carrier-mediated diffusion, and here we see again the use of proteins, but these are called carrier proteins, which imply that the solute has to bind to the protein in order for it to change shape, either to dump it into the cell or out of the cell. We had the facilitated diffusion, which is requiring no energy, but it does require that carrier protein to help it. And then we had active transport, and there it does require energy. Now we can go against the concentration gradient, and we talked about the sodium-potassium exchange pump as our key example. So today's video, this video is over osmosis, which is the diffusion of water. So if we're talking about the diffusion of water, of course we know our term diffusion means movement from a high concentration to a low concentration. And in this case, instead of following the solute, the ions, the sugar, the gases, we're going to follow the water molecule itself. And so our key question here every time is whether our cell is going to gain water, which could cause it to rupture, or lose water, which could cause it to shrivel up, or whether it's going to stay the same. And so that's what we're looking at here. Again, when we're talking about diffusion of water, we're talking about with the concentration gradient from a high to a low concentration. And so this is a passive process. Here is the thing to keep in mind. Water is polar. Likes dissolve likes when it comes to solubility. So water is polar, polar molecules and ions are all going to dissolve in water. We talked about it as the universal solvent. Remember, our term solvent means the dissolver. And so whenever we're going to have this conversation about osmosis and we're talking about water, it's never straight up just pure H2O and that's it. It's water with sodium ion and chloride ion and potassium ion and calcium ion and hydrogen ion and glucose and suspended proteins and all sorts of stuff in with that water. And so it will complicate our conversation quite a bit. Here is our reminder. We have, of course, our solvent is our dissolver, so that was our water, and our solute is the stuff that is dissolved, so we used our lemonade mix as our example there, and of course the solution is referring to the combination of the solute dissolved in the solvent. Again, when we're talking about osmosis, we're talking about the movement of water, whether water enters the cell or leaves the cell, or whether there is the same amount of water moving in and out and the cell stays the same. And so in order to actually have this conversation, we have to look not just at the water itself, but we have to look at the ratio of water and solute in order to determine where we have our most dilute solution and where we have our most concentrated solution. So in order to determine our concentration gradient, we're basically looking at, or we can use the terms dilute and concentrated. The key thing to keep in mind is this ratio here, or relationship I should say. If you have a lot of water in a sample, then you're going to have very little solute in that sample. And so a dilute solution is a watery solution with a lot of water and very little solute. So the reverse of that would be true if you're talking about a concentrated solution, then it's going to have arrows going in the opposite way. So you're going to have low water and high solute in a concentrated solution. What we're trying to do here is just establish the hill. Where's the high water? Where's the low water? Okay. Your dilute solutions are always going to have a higher water concentration. They're going to be at the top of the hill. And our concentrated solutions are always going to have the least amount of water. So they're going to be at the bottom of the hill. So this tells us that water is going to diffuse or move from a high water concentration to a low water concentration, from a dilute solution to a concentrated solution. Okay. Here's where it gets complicated, if you're not already feeling that way. We have some very specific terminology that's used for osmosis. We have this term called tonicity. Tonicity is referring to the solute concentration in the extracellular fluid compared to the intracellular fluid. So every time we're using a tonicity term, we are describing the solute in the ECF compared to the solute in the ICF. So we're interested in which direction water moves, but our terms are defining the solute concentrations. Okay, so let's look at our examples. We have 3 different tonicities. So notice I've made the prefixes large because that is the part of the word that is different: hypertonic, isotonic, and hypotonic. Now I always write isotonic in the middle. Iso is referring to the same. So here the concentration of solutes in the ECF is the same as the concentration of solutes in the ICF. And I've just done this a real basic sort of shorthand here, you know, really super summarized. But here's your basic picture. We have the same number of dots. We have 5 solute inside the cell, 5 solute outside of the cell. So because there is no hill, there is no concentration gradient, it's a flat line, we have the same amount of water going in and out of the cell. So this cell doesn't gain water, it doesn't lose water, it stays the same. So this would be the ideal scenario for animal cells. An isotonic solution. That means the ECF has the same concentration of solute as the ICF, and so the cell doesn't gain or lose water. Now I always write hypertonic above because hypertonic means a high solute concentration in the ECF. This is a salty solution. So notice I still have stuck my 5 solute inside the cell, but I have like so many out here I don't even want to count it. So we could talk about this interstitial fluid as being salty or concentrated, or we can use our proper terminology, it's hypertonic. And so the key thing is that we're going to pull water out of the cell in order to dilute the surrounding solution, and this will cause our cell to lose water. Our cell will dehydrate. We know if we dehydrate, then that's bad, right? That's because your cell is dehydrating first. So if it's bad for your body to be dehydrated as an organism, it's really bad for your cell to be dehydrated as a cell, right? And so this can be very dangerous. Now, this can happen just like I had potato chips for lunch, and then I'm trying to eat my lunch in like 10 minutes. So I'm just shoveling the food in and I'm not drinking a lot. That's why I'm drinking my coffee now. Okay, so I ate some salty lunch and I didn't dilute it out with water. So I'm gonna feel thirsty. I'm gonna have a lot of salt entering into my bloodstream. That salt is gonna enter into my body fluids, and so it's going to make it salty or hypertonic. Well, one of the things that's going to happen is it's going to pull water out of my cells in order to dilute it, but I'm also going to feel thirsty and start drinking more liquids, and then I'm also going to retain water and urinate less in order to maintain that balance. So it's not necessarily, you know, an emergency situation if I eat a salty meal, right? Because my body has mechanisms to maintain homeostasis. Okay. Key thing here, hypertonic solution means you have a higher solute concentration in the ECF compared to the inside of your cell. And so the cell loses water, the cell is dehydrating. The opposite of this is the hypotonic. So I always write this below because hypo means below. So this is a low solute concentration in the ECF. This is where it's super dilute in the ECF and more concentrated inside. So here I have very few yellow dots on the outside, lots of yellow dots on the inside. So now my cell is going to gain water. It's going to swell up, potentially bursting open, and that will then kill my cell. Remember, the term lysis means to burst, so we can talk about the cell lysing. If we're talking about a red blood cell lysing, we call that hemolysis, where heme prefix means blood. So this can happen if you're drinking excessive amounts of water and you're not replacing or balancing that out with electrolytes, so you can kind of flood your body fluids, and then that can to some extent flood your cells because water will move into those cells. Luckily, we have mechanisms in the kidney to help pull off that extra water. So if you're drinking excessive amount of water, then you're going to urinate frequently as you're pulling that water out of the body. If you drink excessive amounts of water in a really short period of time, you don't have enough time to actually process and remove that extra water from your body. And so that can actually cause you to be sick in the same way that dehydration can cause you to be sick. And ultimately, it can kill you. So you have to be careful about that. Okay, so tonicity was talking about the amount of solute in this ECF compared to the ICF. So it was a comparison term, and it was always specific to the extracellular fluid. This term, osmolarity, can be used to describe any solution, and it's just describing the total amount of solute in the solution. So how salty that solution is. So the term to describe if you have a salty solution, you can call it a concentrated solution. You can say it has a high osmolarity. So in the case of our hypertonic solution, we can say it had a high osmolarity. If your solution is dilute, then we can describe that as a low osmolarity. That just means fewer salts. So a hypotonic solution has a lower osmolarity compared to the cell. And that is it for now. So stay tuned for our last video in part 1.